Extracellular polymeric substances, EPS, are complex mixtures of polymers, primarily polysaccharides, proteins, lipids, and nucleic acids produced by microbial cells. They form a matrix that surrounds cells in biofilms, providing structural stability, protection against environmental stress, and facilitating nutrient retention and microbial communication. Mycoplasms, on the other hand, are a unique group of bacteria that lack a cell wall, making them resistant to antibiotics like penicillin that target peptidoglycan synthesis. Their flexibility and small genome enable them to adapt to various environments as parasitic or saprophytic organisms. Ribosomes, the sites of protein synthesis, differ between prokaryotes and eukaryotes. Prokaryotic ribosomes are 70S, composed of 50S and 30S subunits, while eukaryotic ribosomes are larger, 80S, consisting of 60S and 40S subunits. These structural differences are exploited in antibiotic development, as many drugs selectively target prokaryotic ribosomes without affecting eukaryotic. The cytosol and cytoskeleton are essential components of a cell's internal structure. The cytosol is the gel-like fluid that fills the intracellular space, supporting organelles and facilitating the movement of molecules and ions necessary for cellular processes. Within the cytosol lies the cytoskeleton, a dynamic network of protein filaments, microtubules, actin filaments, and intermediate filaments that maintain cell structure, enables extracellular transport, and assist in cell division and signaling. Chromosomes, the carriers of genetic information, are highly organized structures of DNA and proteins, primarily histones, forming a complex called chromatin. In eukaryotic cells, chromosomes condense during cell division to ensure accurate distribution of genetic material. The endosymbiotic theory explains the evolutionary origin of eukaryotic cells, proposing that mitochondria and chloroplasts originated as free-living prokaryotes engulfed by an ancestral eukaryotic cell. This mutual beneficial relationship eventually led to those organelles becoming integral components of eukaryotic cells, evidenced by their double membrane, circular DNA, and similarities to prokaryotic ribosomes. The reaction rate of biochemical processes is significantly influenced by factors such as temperature, pressure, and the presence of enzymes. Enzymes, which are proteins with specific three-dimensional structures, act as a biological catalyst to speed up reactions by lowering activation energy. Temperature affects enzyme activity by increasing molecular movement, but excessive heat can denature enzymes, disrupting their structure and function. Pressure can also impact reaction rates, particularly in systems involving gases, as it alters molecular interactions and concentrations. Lipid structures consist of hydrophobic tails and hydrophilic heads, are broken down by lipases, enzymes that hydrolyze triglycerides into glycerol and fatty acids. Similarly, protein structure stabilized by peptide bonds and hydrogen bonds and disulfide bonds is targeted by proteases and peptidases, which cleave proteins into smaller peptides or amino acids. These enzyme-mediated reactions are highly sensitive to environmental conditions, showcasing the intrinsic interplay between molecular structure and function in a living system. Don't forget to like, comment, share, and subscribe to this channel to get more retention videos. I'll see you in the next video.